How do animals and people identify smells?
The sense of smell allows animals and humans as well as other organisms to identify food, mates, and predators. This sense also provides sensory pleasure, e.g., flowers, and warnings of danger, example chemical dangers. There are specialized receptor cells in the nose that have proteins that bind chemical odorants and cause the receptor cells to send electrical signals to the olfactory bulb of the brain. Cells in the olfactory bulb relay this information to olfactory areas of the forebrain to generate perception of smells.
What is nuclear magnetic resonance?
Nuclear magnetic resonance, NMR, is a process in which the nuclei of certain atoms absorb energy from an external magnetic field. Scientists use NMR spectroscopy to identify unknown compounds, check for impurities, and study the shapes of molecules. They use the knowledge that different atoms will absorb electromagnetic energy at slightly different frequencies.
What were the smallest and largest dinosaurs?
Compsognathus, a carnivore from the late Jurassic period, 131 million years ago, was about the size of a chicken and measured, at most, 35 in, 89 centimeters, from the tip of its snout to the tip of its tail. The average weight was about 6.8 pounds 3 kilograms, but individuals could be as much as 15 pounds 6.8 kilograms. The largest species for which a whole skeleton is known is Brachiosaurus. A specimen in Humboldt Museum in Berlin measures 72.75 feet 22.2 meters long and 46 feet 14 meters high. It weighed an estimated 34.7 tons, 31,480 kilograms. Brachiosaurus was a four-footed plant-eating dinosaur with a long neck and tail and lived from about 155 to 131 million years ago.
What is an exon?
An exon is a segment of the transcript that is tied together with other exons by the spliceosome to make the mRNA molecule. After the exons are spliced together, they exit the nucleus for the cytoplasm, where the mRNA is translated into a polypeptide.
Which fungi commonly found in North America have demonstrated beneficial medicinal effects?
Clinical trials have been conducted to test potential health benefits of fungi commonly found in North America. The following chart reveals some of the benefits that several common fungi may provide.
What is the importance of the rainforest?
Half of all medicines prescribed worldwide are originally derived from wild products. And the United States National Cancer Institute has identified more than 2,000 tropical rainforest plants with the potential to fight cancer. Rubber, timber, gums, resins and waxes, pesticides, lubricants, nuts and fruits. Flavorings and dyes, tufts, steroids, latexes, essential and edible oils, and bamboo are among the products that would be drastically affected by the depletion of the tropical forests.
What genetic diseases come from mutations of mitochondrial DNA?
Because mitochondria are primarily responsible for energy conversion. Mutations in their DNA affect the ability of the cell to generate ATP, the immediate energy source for cells. Those cells in tissues with the highest energy demands demonstrate the most severe effects of mitochondrial mutations.
Was the Exxon Valdez spill the largest oil spill of the 20th century?
Although the Exxon Valdez was widely publicized as a major spill of 255,500 barrels, 35,000 tons, in 1989, it was not the largest of the century. The first major commercial oil spill occurred on March 18, 1967, when the tanker Torrey Canyon grounded on the Seven Stones Shoal off the coast of Cornwall, England, spilling 830,000 barrels, 119,000 tons, of Kuwaiti oil into the sea. This was the first major tanker accident. However, during World War II, German U-boat attacks on tankers between January and June 1942 spilled 4.3 million barrels, 590,000 tons, of oil off the east coast of the United States. Even this spill is dwarfed by the deliberate dumping of oil from Sea Island into the Persian Gulf during the First Gulf War in 1991. It is estimated that the Sea Island spill equaled almost 10.9 million barrels, 1.5 million tons, of oil. A major spill also occurred in Russia in October 1994 in the Komi region of the Arctic. The size of the spill was reported to be as much as 2 million barrels, 286,000 tons. In addition to the large disasters, day-to-day pollution occurs from drilling platforms where waste generated from platform life, including human waste, and oils, chemicals, mud, and rock from drilling are discharged into the water.
Where are bacteria found?
Bacteria inhabit every place on Earth, including places where no other organism can survive. Bacteria have been detected as high as 20 miles 32 kilometers above the Earth and 7 miles 11 kilometers deep in the waters of the Pacific Ocean. They are found in extreme environments, such as the Arctic tundra, boiling hot springs, and our bodies.
How can animals be grouped according to type of body cavity?
The structures of most animals develop from 3 embryonic tissue layers. The outer layer, the ectoderm, gives rise to the outer covering of the body and the nervous system. The inner layer, the endoderm, forms the lining of the digestive tube and other digestive organs. The middle layer, the mesoderm, gives rise to most other body structures, including muscles, skeletal structures, and the circulatory system. One system of grouping animals with 3 germ layers, triploblastic, is based on the presence and type of body cavity. Or chylome, a fluid-filled space between the body wall and digestive tube. Achylomates such as flatworms, phylum Platyhelminthes, have a solid body with no body cavity. Pseudocoelomates have a false chylome, since the body cavity is not completely lined with mesoderm. Pseudocoelomates include such animals as roundworms, phylum Nematoda, and Rotifers, phylum Rotifera. Coelomates, which have a body cavity that is completely lined with mesoderm, include most other animals ranging from earthworms, phylum Annelida, to vertebrates, phylum Chautata.
What is the cell cycle and how is it controlled?
The life cycle of a single eukaryotic cell is known as the cell cycle. The cycle has 2 major phases, interphase and mitosis. When a cell is not dividing, it is in interphase, for example. A mature neuron conducting an impulse is in interphase. Some cells remain in interphase almost indefinitely. Interphase includes the G1, S, and G2 phases, which are periods of growth during which a cell increases in size, complexity, and protein content. The G1 phase prepares for DNA synthesis, known as the S phase. And G2 prepares for both mitosis and the synthesis of proteins. Cells that undergo mitotic phases of division and renewal are cyclically predictable. An example of this would be an epithelial cell, which divides about every 15 days. The phase of mitosis, also known as M phase, may require a great deal of preparation time.
What does the word amphibian mean?
The word amphibian, from the Greek term amphibia, means both lives and refers to the animal's double life on land and in water. The usual life cycle of amphibians begins with eggs laid in water. which develop into aquatic larvae with external gills, in a development that recapitulates its evolution. The fish-like larva develops lungs and limbs and becomes an adult.
What is a chromosome map?
A chromosome map lists the sequence of genes found on a given chromosome. Chromosome maps are usually determined by breeding experiments in which the ratio of the offspring with certain combinations of traits indicates how far apart those traits are on the chromosome.
Is influenza a serious viral disease?
The influenza virus is probably the most lethal virus in human history. It is an acute respiratory disease characterized by fever, chills, headache, generalized muscular aches, and a frequent cough. Influenza affects people of all ages, but can be particularly severe for the very young, the very old, and people with complications due to other diseases. It is estimated that within 18 months during 1918 to 1919, there were 200 million cases of influenza, causing 21 million deaths worldwide.
What is a daddy longlegs?
The name applies to two different kinds of invertebrates. The first is a harmless, non-biting, long-legged arachnid. Also called a harvestman, it is often mistaken for a spider. But it lacks the segmented body shape that a spider has. Although it has the same number of legs, 8. As a spider, the harvestman's legs are far longer and thinner. These very long legs enable it to raise its body high enough to avoid ants or other small enemies. Harvestmen are largely carnivorous, feeding on a variety of small invertebrates such as insects, spiders, and mites. They never spin webs as spiders do. They also eat some juicy plants and in captivity can be fed almost anything edible, from bread and milk to meat. Harvestmen also need to drink frequently. The term daddy longlegs also is used for a cranefly, a thin-bodied insect with long thin legs that has a snout-like proboscis with which it sucks water and nectar.
What are some examples of modified leaves?
Some leaves are modified to perform functions other than photosynthesis, while the tendrils of some plants are modifications to the stems and provide support for the plant. In other species, such as pea plants, Pisum sativum, the tendrils are modified leaves. In carnivorous plants, such as the Venus flytrap, Dionaea muscipula, and the pitcher plant, Sarracenia purpurea, the leaves attract, capture, and digest the insects with enzymes. The leaves of many desert plants grow mainly underground with only a small transparent window tip protruding above the soil surface. The soil covering the leaf protects it from dehydration by the harsh desert winds. The window allows light to penetrate and reach the site of photosynthesis.
What is bDNA?
There are three different structural forms of DNA. The secondary structure of DNA varies according to the amount of water surrounding the molecule. The structure that Watson and Crick described is bDNA. And evidence supports that this is the predominant form of most cells. bDNA is a beta helix with a clockwise spiral. Another structural form, aDNA, exists when there is less water immediately surrounding the molecule. aDNA is an alpha, right-handed, helix, but it is shorter and wider than bDNA. The third form, zDNA, is a left-handed helix in which the sugar phosphate groups zigzag. This form is found when certain specific base sequences are present.
What is the most dangerous poison produced by a plant?
In North America, the poisonous hemlock, Conium maculatum, is probably the most dangerous plant. The South American lana tree is another dangerous plant. Native Americans used its sap to make curare, a poisonous substance applied to their arrows and spears. It can cause death in a matter of minutes.
What are buds on stems?
Buds may be terminal or axillary on a stem. The terminal bud is at the apex of the stem where the plant growth is concentrated. It contains developing leaves and a compact series of nodes and internodes. The axillary buds are found in the angles formed by a leaf and the stem. The axillary buds are usually dormant.
How can animal thought processes be studied?
Determining how an animal thinks will involve scanning the brain of the animal while performing a cognitive task. Data would then be compared with humans. Although human functional MRI experiments are becoming common, such experiments on other animals are still quite rare.
Will wild birds reject baby birds that have been touched by humans?
No. Contrary to popular belief, birds generally will not reject hatchlings touched by human hands. The best thing to do for newborn birds that have fallen or have been pushed out of the nest is to locate the nest as quickly as possible and gently put them back.
What three groups are included in the flatworms?
Flatworms belong to the phylum Platyhelminthes. They are flat, elongated, achylomate animals that exhibit bilateral symmetry and have primitive organs. The members of the flatworms are 1. Planarians, 2. Flukes, and 3. Tapeworms.
What is a bar body?
In 1949 Dr. Murray Barr, 1908 to 1995, noticed a dark body in the neurons of female cats. It was later identified as a structure found only in the nucleus of females. It was named a Barr body in honor of its discoverer.
What are chaperone proteins?
While most proteins fold spontaneously into their specific conformation, some require the help of other molecules to achieve this. Chaperone proteins guide newly synthesized polypeptides into their functional three-dimensional shape.
How do the great whales compare in weight and length? What is the fastest swimming whale?
The orca or killer whale, or Sinis orca, is the fastest swimming whale. In fact, it is the fastest swimming marine mammal with speeds that reach 31 miles per hour, 50 kilometers per hour.
What is acid rain?
The term acid rain was coined by the British chemist Robert Angus Smith, 1817-1884, who in 1872 published Air and Rain, The Beginnings of a Chemical Climatology. Since then, acid rain has unfortunately become an increasingly used term for rain, snow, sleet, or other precipitation that has been polluted by acids such as sulfuric and nitric acids. When gasoline, coal, or oil are burned, their waste products of sulfur dioxide and nitrogen dioxide combine in complex chemical reactions with water vapor in clouds to form acids. The United States alone discharges 40 million metric tons of sulfur and nitrogen oxides into the atmosphere. This, combined with natural emissions of sulfur and nitrogen compounds, has resulted in severe ecological damage. Hundreds of lakes in North America, especially in northeastern Canada and the United States, and in Scandinavia, are so acidic that they cannot support fish life. Crops, forests, and building materials such as marble, limestone, sandstone, and bronze have been affected as well, but the extent is not as well documented as it is with fish life. However, in Europe, where many trees are stunted or have been killed, a new word Waldsterben, forest death, has been coined to describe this phenomenon. In 1990 amendments to the U.S. Clean Air Act contained provisions to control emissions that cause acid rain. The amendments called for the reductions of sulfur dioxide emissions from 19 million tons to 9.1 million tons annually and the reduction of industrial nitrogen oxide emissions from 6 to 4 million tons annually.
Do plant cells really produce oxygen?
Yes, plant cells produce oxygen through the process of photosynthesis. Splitting water molecules to harvest their electrons causes the release of oxygen. By submerging a small piece of an aquatic plant in a beaker containing water, one can actually see the oxygen bubbles produced.
Which bacteria were used to clean the beach after the Exxon Valdez oil spill?
B. Bacteria from the genus Pseudomonas are capable of degrading oil as their source of nutrition. This microorganism was used to clean up the spill that emerged from the Exxon Valdez. In order to improve the efficiency of this bacteria, nitrogen and phosphorus were introduced to beaches polluted by the spill. The number of oil-degrading bacteria on the beaches increased sufficiently enough to remove much of the oil from the beaches.
What plants can be used to determine blood type?
Lectins, proteins that bind to carbohydrates on cell surfaces found in lotus plants, as well as jack and lima beans, can be used to determine a person's blood type. Lectins bind to glycoproteins present on the plasma membrane of red blood cells. Because the cells of different blood types have distinct glycoproteins, cells of each blood type bind to a specific lectin.
What is the alternation of generations in plants?
All plants exhibit an alternation of generations between diploid sporophytes and haploid gametophytes. Sporophytes produce haploid spores as a result of meiosis. The spores grow into multicellular, haploid individuals known as gametophytes. Spores are the first cells of the gametophyte generation. Gametophytes produce gametes as a result of mitosis. Male and female gametes fuse to form a zygote, which grows into a sporophyte. The zygote is the first cell of the following sporophyte generation.
What is a knockout mouse?
A knockout mouse is a mouse that has specific genes mutated, knocked out, so that the lack of production of the gene product can be studied in an animal model. Knockout mice are frequently used in pharmaceutical studies to test the potential for a particular human enzyme as a therapy target. Mice have identical or nearly identical copies of human proteins, making them useful in studies that model effects in humans.
What is the status of the African elephant?
From 1979 to 1989, Africa lost half of its elephants from poaching and illegal ivory trade, with the population decreasing from an estimated 1.3 million to 600,000. This led to the transfer of the African elephant from threatened to endangered status in October 1989 by CITES, the Convention on International Trade in Endangered Species. An ivory ban took effect on January 18, 1990. Botswana, Namibia, and Zimbabwe have agreed to restrict the sale of ivory to a single, government-controlled center in each country. All countries have further pledged to allow independent monitoring of the sale, packing, and shipping process to ensure compliance with all conditions. Finally, all three countries have promised that all net revenues from the sale of ivory will be directed back into elephant conservation for use in monitoring, research, law enforcement, other management expenses, or community-based conservation programs within elephant range.
What is a calorie?
There are actually two kinds of calories. Ask a chemist and you will learn that a calorie, with a lowercase c, is the amount of energy, heat, required to raise 1 gr, 1 ml, of water by 1 degree Celsius. A nutritionist, on the other hand, would describe a big C or kilocalorie. Kcal, the amount of energy required to raise 1 kilogram, 1 L, of water by 1 degree Celsius. The kcal is the unit used to describe the energy value in food. For example, if a chocolate chip cookie is completely incinerated, the amount of heat energy released would be enough to raise the temperature of 1 L of water by approximately 300 degrees Celsius. However, as this system adheres to the laws of thermodynamics, the conversion is not totally efficient. Only about 25% of energy actually performs cellular work.
Why is electrophoresis important for biological research?
Gel electrophoresis has become an important tool to identify DNA molecules, genetic differences between different species of plants, and animals have been identified using gel electrophoresis. Protein molecules that differ by a single amino acid can be identified. This process is useful for determining the genetic fingerprint of an individual.
How do conservationists predict whether a species will become extinct?
Conservation biologists use population viability analysis, PVA, a relatively new method, to predict the viability of a species in a particular habitat. Computer modeling generates PVAs using life history data, genetic variability, and a population's response to environmental conditions, especially disturbances, to predict viability of a species.
Why do animals play?
Many animals, mammals and some birds, particularly, have been observed at play during different stages of development. Although play would seem to be random, ethologists have described three patterns: 1. Social. For establishing relationships to other animals; 2. Exercise, for development of muscles; 3. Exploration of an object. All of these occur during the wrestling, chasing, and tumbling activities of the young of many species. Some juvenile play, such as lion cubs capturing mice, may be practice for adult activities such as hunting.
When were chromosomes first observed?
Chromosomes were observed as early as 1872, when Edmund Rousseau, 1841 to 1897, described seeing items that resembled small rods during cell division. He named the rod stab chen. Edouard van Beneden, 1846 to 1910, used the term bâtonnet in 1875-40 to describe nuclear duplication. The following year, 1876, Edouard Balbiani, 1825 to 1899, described that at the time of cell division the nucleus dissolved into a collection of bâtonnets centroids, narrow little rods. Walther Fleming, 1843 to 1905, discovered that the chromosomal threads or phaeton split longitudinally during mitosis.
What are some applications of gas chromatography?
Gas-liquid chromatography is the most widely used chromatographic technique for environmental analyses. Analysis of organic compounds is possible for a variety of matrices such as water, soil, soil gas, and ambient air. It is often used to test hazardous waste sites for determining personal protective equipment. PPE, level and emergency response testing.
How is it possible to identify the genus and species of a plant based on its wood?
The arrangement of plant cells present in the xylem of Wood allows for the identification of a plant's genus. For example, the wood of oak trees, genus Quercus, is dense and heavy because it has abundant fibers.
What is torpor?
Torpor is a short-term decrease in body temperature and metabolic rate. Animals such as hummingbirds and bats go through daily periods of torpor that allow them to reduce their energy requirements at night or when hunting is poor. Torpor can be considered as a type of brief sleep, but it is distinct from a state from hibernation.
What is flow cytometry?
Flow cytometry is an exciting new biotechnology method in which single cells or subcellular fractions flowing past a light source are scanned based on measurement of visible or fluorescent light emission. The cells can be sorted into different fractions depending on fluorescence emitted by each droplet. Flow cytometry can be used to identify particular cell types such as malignant cells or immune cells such as T or B cells. High-throughput screening is a large-scale automated search of a genome in order to determine how a cell or larger biological system uses its genetic information to create a specific product or total organism. The term platform is used to describe all components needed for high-throughput screening.
Why don't organisms that produce toxins die during the synthesis of toxic material?
Organisms produce toxins as a defense mechanism, therefore, most organisms are immune to their own toxins as well as that of other members of the same species. Toxins are a cell product packaged into a membranous sac known as a vesicle. The contents of the vesicle are secreted into the salivary gland, skin surface, or fangs of the organism during the process of exocytosis. The toxin is then ingested, or absorbed by other organisms, often a predator. Each toxin creates a specific effect. For example, the snake venom known as bungarotoxin deactivates acetylcholine receptors on muscles. Since it is secreted externally, it is unlikely that the snake producing the toxin would be affected.
What is camouflage behavior?
Camouflage behavior allows an animal to avoid predation by appearing to be something it is not. Examples of this include the behavior known as freezing. In addition to the opossum, hog-nosed snakes also exhibit this behavior. Freezing may at first seem counterproductive as an anti-predator strategy. But it turns out that some predators, e.g., frogs and wolves, will not attack a non-moving target.
What is habituation?
Habituation is the decreased response to a stimulus that is repeated without reinforcement. Habituation can be very important to an animal in its natural surroundings. As an example, young ducklings run for cover when a shadow, a possible predator, passes overhead. Gradually, however, the ducklings learn which types of shadows are dangerous and which are harmless.
Is lactose intolerance a genetic disorder?
Lactose intolerance is due to reduced activity of the enzyme lactase. Lactase hydrolyzes the lactose of mammalian milk into the two monosaccharides glucose and galactose. Lactose intolerance is very common in adults, as expression of the lactase enzyme begins to decrease in humans at about 2 years of age. Estimates of lactose intolerance in the U.S. population vary. Congenital lactase deficiency is a rare autosomal recessive disorder and results in no lactase production. This is in contrast to childhood and adult onset lactase deficiency, which is common and is inherited in an autosomal dominant pattern.
How do some cells, such as those found in nails and horns, change shape and become hard?
All of the surface cells of the body, except those in eyes, contain a molecule called keratin, a fibrous protein that is particularly well suited to withstand abrasions. Certain cells, like those of the nails and hair, have increased amounts of keratin that provide extra strength, helping them maintain their shape. Whether it is in the claws on the toes of your cat, the horns on the head of your favorite cow, or the 5 million hairs that cover your body, keratin strengthens and protects organisms from everyday wear and tear.
What is kudzu?
Kudzu, pure Aria lobata, is a vine that was brought from Japan for the 1876 Centennial Exposition in Philadelphia. It was intentionally planted throughout the southern United States during the 1930s in an attempt to control erosion. In fact, the federal government paid farmers as much as $8 an acre to plant it. In 1997, however, the government reversed its position on kudzu and referred to it as a noxious weed. Kudzu grows over everything that it encounters, draping itself across power poles and pine trees like a shawl. The plant is responsible for more than $50 million in lost farm and timber production each year. It grows at a rate of 120,000 acres per year. As of the early 21st century, it covers between 2 and 4 million acres of land throughout the United States, occurring from Connecticut in the east to Missouri and Oklahoma in the west, and south to Florida and Texas. Kudzu grows as fast as 1 foot 30 centimeters per day. The latest approach to controlling the growth of kudzu is to have goats chew on it, devouring the leaves, stems, and roots.
What is a chromosome?
A chromosome is the thread-like part of a cell that contains DNA and carries the genetic material of a cell. In prokaryotic cells, chromosomes consist entirely of DNA and are not enclosed in a nuclear membrane. In eukaryotic cells, the chromosomes are found within the nucleus and contain both DNA and RNA.
What is Gray's manual?
Gray's Manual of Botany, first published in 1848 by Asa Gray, 1810 to 1888, under the title Manual of the Botany of Northern United States, was the first accurate and modern guide to the plants of eastern North America. The publication contained keys and thorough descriptions of plants. The 8th, and Centennial, edition was largely rewritten and expanded by Merritt Lyndon Fernald. 1873 to 1950, and published in 1950. This edition was corrected and updated by R.C. Rollins and reprinted in 1987 by Dioscorides Press.
What is lyophilization?
Lyophilization is a freeze-drying technique for preservation and storage of bacteria and other microorganisms. Bacteria can be stored for extended periods of time, 3 to 5 years, as frozen cell suspensions or as freeze-dried, lyophilized, cultures. This technique is achieved by placing bacteria in a nutrient broth containing 15 to 25% glycerol and freezing at temperatures of -94 degrees Fahrenheit -70 degrees Celsius or lower. The glycerol reduces ice crystal formation that would cause subsequent cell damage and disrupt biological structures.
What are mycoplasmas?
Mycoplasmas are the smallest, free-living organisms, and the only bacteria that exist without a cell wall. Some mycoplasmas have sterols, a type of lipid-lacking fatty acids, in their plasma membranes, which provide the strength a membrane needs to maintain cellular integrity without a wall. Since mycoplasmas lack cell walls that provide shape and rigidity, they have no definitive forms and are pleomorphic. Mycoplasma pneumoniae causes a disease known as primary atypical pneumonia. PAP, a mild form of pneumonia confined to the lower respiratory tract. Because mycoplasmas do not have cell walls, penicillin is ineffective in stopping their growth. Tetracycline, which inhibits protein synthesis, is recommended as the antibiotic of choice for treatment of PAP.
What are the two distinct body forms of cnidarians?
The two forms are called the polyp stage and the medusa, plural, medusae, or jellyfish, stage. Polyps generally live attached to a hard surface and bud to produce more polyps and, in some cnidarians, to produce the medusa stage of the life cycle. These medusae, or jellyfish, drift with the ocean currents or swim by pulsating their umbrella-shaped bodies. Medusae release sperm and eggs into the water, where external fertilization occurs. After fertilization, the embryo develops into a larva that eventually settles to the bottom to become another polyp, thus completing the life cycle. Not all cnidarians go through both polyp and medusa stages. Some, such as corals and sea anemones, exist only as polyps.
How many eggs does a spider lay?
The number of eggs varies according to the species. Some larger spiders lay more than 2,000 eggs. But many tiny spiders lay 1 or 2 and perhaps no more than a dozen during their lifetime. Spiders of average size probably lay 100 or so. Most spiders lay all their eggs at one time and enclose them in a single egg sac. Others lay eggs over a period of time and enclose them in a number of egg sacs.
Do all brain cells work the same way?
There are two basic types of cells in the nervous system: neurons that carry messages and the support cells that both feed and protect them. In the central nervous system comprised of the brain and spinal cord, these support cells are known as neuroglial or glial cells. These cells perform a variety of functions in maintaining the health of the neurons of the brain. It is estimated that glial cells actually comprise about 90% of the cells in the nervous system.
How is commercial cork cultivated?
Commercial cork is the outer bark of the cork oak, Quercus suber, grown in the western Mediterranean. The first periderm is commercially useless. It is removed from the tree and discarded when the tree is approximately 10 years old. When the tree is 20 to 25 years old and has a diameter of approximately 15.75 in 40 centimeters, a usable cork layer 1.2 to 3.9 in 3 to 10 centimeters thick can be harvested. A similar layer can be harvested approximately once every 10 years until the tree is approximately 150 years old. The cork of a cork oak breaks away at the cork cambium and can be peeled off without harming the tree.
What are vitamins and minerals?
A vitamin is an organic, non-protein substance that is required by an organism for normal metabolic function but cannot be synthesized by that organism. In other words, vitamins are crucial molecules that must be acquired from outside sources. While most vitamins are present in food, vitamin D, for example, is produced as a precursor in our skin and converted to the active form by sunlight. Minerals, such as calcium and iron, are inorganic substances that also enhance cell metabolism. Vitamins may be fat or water-soluble. Recommended amounts of vitamins are to ensure normal enzymatic function, and excessive intake can be toxic.
How often is the epithelium replaced?
Epithelial cells are constantly being replaced and regenerated during an animal's lifetime. The epidermis, outer layer of the skin, is renewed every 2 weeks, while the epithelial lining of the stomach is replaced every 2 to 3 days. The liver, a gland consisting of epithelial tissue, easily regenerates after portions are removed surgically.
How do animals mark their territory?
Animals use pheromones to mark their territory. A pheromone is a type of airborne chemical. This can be accomplished by leaving bits of fur and dander on a visual object or by scent marking with small amounts of urine.
How does an animal's expression reflect behavior?
Although only primates have the facial musculature to be truly expressive, many species use their appearance to convey information. For example, pufferfishes expand their bodies to look more threatening, as do dogs and cats when they raise their hackles and puff up their body fur. Cats, horses, and others also use their ears to relay information about their intentions. And certainly, many a pet owner has learned to read his dog's mood by the movement of the tail. Unlike female humans, most female mammals have an estrous cycle instead of a menstrual one. Around the time of ovulation, these females experience estrus, a period of sexual receptivity. Sexual readiness can be advertised in a variety of ways, including physical and behavioral changes. Among females in species with estrus, it is common to see a swelling of the external genitalia. Female chimpanzees and other primates demonstrate this phenomenon. Male mandrills exhibit sexual maturity and dominance by the vibrancy of the coloration on the facial ridges and posterior flesh. When male elephants reach sexual maturity, they experience must, a condition typified by leakage of fluid from the penis as well as oozing of tears from the face. Of course, members of both sexes are likely to also exhibit changes in behavior. actively seeking out the opposite sex, for example. Cats and dogs in heat will go to extraordinary lengths to meet a potential mate.
When was the Scopes monkey trial?
John T. Scopes, 1900 to 1970, a high school biology teacher, was brought to trial by the state of Tennessee in 1925 for teaching the theory of evolution. He challenged a law passed by the Tennessee legislature that made it unlawful to teach in any public school any theory that denied the divine creation of man. He was convicted and sentenced, but the decision was reversed later and the law repealed in 1967. In the early 21st century, pressure against school boards still affects the teaching of evolution. Recent drives by anti-evolutionists either have tried to ban the teaching of evolution or have demanded equal time for special creation as described in the biblical book of Genesis. This has raised many questions about the separation of church and state. The teaching of controversial subjects in public schools, and the ability of scientists to communicate with the public. The gradual improvement of the fossil record, the result of comparative anatomy, and many other developments in biological science has contributed toward making evolutionary thinking more palatable.
How do genes control cancer cells?
In normal cells, there are two types of genes that are important in determining whether or not cancerous tissue can form. These genes control the production of proteins that affect the cell cycle. Proto-oncogenes are DNA sequences that promote normal cell division. By mutation, these genes may be converted into oncogenes, which promote the overproduction of cells. Another class of genes, known as tumor suppressor genes, prevents excess reproduction of cells. Mutation in these genes can also allow cells to become cancerous.
What is a transposon?
A transposon is a nucleotide sequence in a gene that literally jumps around from one chromosome to another. Transposons can be problematic, as they may disrupt the normal function of an important gene by their random insertion into the middle of that gene. However, transposons are found in almost all organisms, and their presence in a genome indicates that genetic information is not fixed within the genome. By studying results of the Human Genome Project, scientists predict that about 45% of all human genes may be derived from transposons.
What is a homeobox gene?
The homeobox is specific subset of nucleotides in homeotic Genes that have important roles in embryonic development. Homeotic genes code for proteins that act as transcription factor, enhancing the rate at which certain DNA sequences are transcribed. The similarity of the sequence in different groups of species is another indication of the shared evolutionary history of species.
Are viruses living organisms?
Viruses cannot grow or replicate on their own and are inert outside their living host cell. Once they enter a host cell, they become active. As such, they are between life and non-life and are not considered living organisms.
What are the commercial uses of brown algae?
Kelp, one of the species of brown algae, is harvested from ocean water for the sodium, potassium salts, iodine, and alginates that it contains. The alginates are carbohydrates used in the formation of gels.
How do chromoplasts differ from chloroplasts?
Chromoplasts are pigment-containing plastids found in plants. They are responsible for the characteristic red, orange, or yellow coloration visible in some flowers, fruits, and plants.
What is the most famous roundworm?
One soil nematode, Caenorhabditis elegans, is widely cultured and has become a model research organism in developmental biology. The study of this animal was begun in 1963 by Sidney Brenner, 1927, who received the Nobel Prize in Physiology or Medicine in 2002. The species normally lives in soil but is easily grown in the laboratory in petri dishes. It is only about 0.06 in, 1.5 millimeters, long, has a simple transparent body consisting of only 959 cells, and grows from zygote to mature adult in only 3.5 days. The genome, genetic material, of C. elegans, consisting of 14,000 genes, was the first animal genome to be completely mapped and sequenced. The small transparent body of this nematode allows researchers to locate cells in which a specific developmentally important gene is active. These cells show up as bright green spots in a photograph because they have been genetically engineered to produce a green fluorescent protein known as GFP. The complete wiring diagram of its nervous system is known, including all the neurons and all connections between them. Much of the knowledge of nematode genetics and development gained from the study of C. elegans is transferable to the study of other animals.
How do diet drugs affect cell metabolism?
Diet drugs target the areas of the brain that control food intake and produce neurotransmitters. Drugs that attempt to control food intake are adrenergic. Their role is to enhance metabolism and increase the efficiency of calorie burning. The second type of drug targets the brain chemical serotonin. Serotonin is linked to appetite, particularly cravings for carbohydrates. Higher levels of serotonin lead to less cravings for carbohydrates. For example, the diet drug dexvenfluramine inhibits the removal of serotonin in the brain. However, there are side effects to the use of diet drugs, and there is insufficient data regarding the safety and effectiveness of their long-term use.
How are cilia and flagella similar?
Cilia and flagella are the motile appendages of eukaryotic cells. They are thick, flexible structures that exhibit a beating motion and project from the surfaces of many cells. A cell having one or a small number of appendages can be identified having flagella. Singular, flagellum, if they are relatively long in proportion to the size of the cell. If the cell has many short appendages, they are called cilia, singular, cilium. Cilia and flagella have the same internal structure, but differ in their length. The number occurring per cell, and mode of beating. They are axon-emal in shape, formed by a main cylinder of tubules that are approximately 0.25 in diameter. The axoneme has a pattern of 9+2 consisting of 9 outer doublets of tubules and 2 additional central microtubules, the central pair. Cilia are about 2 to 10 m long, while flagella are much longer, ranging from 1 m to several millimeters, although they are most 50 commonly 10 to 200. They are both intracellular structures, bound by an extension of the plasma membrane. Both cilia and flagella are used by cells to move through watery environments or to move materials across cell surfaces.
How does a nematocyst work?
A nematocyst is a specialized organelle found in all cnidarians. Each nematocyst features a coiled, thread-like tube lined with a series of barbed spines. The nematocyst is used to capture prey and may also be used for defense purposes. When it is triggered to discharge, the extremely high osmotic pressure within the nematocyst, 140 atmospheres, causes water to rush into the capsule, increasing the hydrostatic pressure and expelling the thread with great force. The barb instantly penetrates the prey, stinging it with a highly toxic protein.
Who first demonstrated that blood circulates?
William Harvey, 1578 to 1657, was the first person to demonstrate that blood circulates in the bodies of humans and other animals. Harvey's hypothesis was that the heart is a pump for the circulatory system, with blood flowing in a closed circuit. Harvey conducted his research on live organisms as well as dissection of dead organisms to demonstrate that when the heart pumps, blood flows into the aorta. He observed that when an artery is slit, all the blood in the system empties. Finally, Harvey demonstrated that the valves in the veins serve to return blood to the heart.
What gases are commonly used for the mobile phase of gas chromatography?
The carrier gas must be chemically inert. Commonly used gases include nitrogen, helium, argon, and carbon dioxide.
Are there other medicinal uses for fungi?
Two species of fungi have been popular in China and Japan for their medicinal value: Lentinula idodes, a species of shiitake, and Ganoderma lucidum, a reishi mushroom. Lentinan and L. idodes mycelium extract, LEM, are two extracts from L. idodes that are being studied for their medicinal properties. Lentinan has been found to enhance the function of the immune system and slow tumor growth. LEM appears to improve liver function and may have potential as an antiviral agent. Asian herbalists consider reishi to be the fungus of choice for treating of a variety of illnesses.
What is DNA?
Deoxyribonucleic acid, DNA, is the genetic material for all cellular organisms. The discovery of DNA is considered the most important molecular discovery of the 20th century.
What are the parts of a flower?
In a generalized flower, there are four main parts. Sepals found on the outside of the bud or on the underside of the open flower. They serve to protect the flower bud from drying out. Some sepals ward off predators by their spines or chemicals. Collectively, the sepals form the calyx. Petals serve to attract pollinators and are usually dropped shortly after pollination occurs. Collectively, the petals form the corolla. Stamen, the male part of a flower, consists of a filament and anther where pollen is produced. Pistil, the female part of a flower, consists of the stigma, style, and ovary containing ovules. After fertilization, the ovules mature into seeds. If a flower has all of these parts, it is called complete. If it lacks any of them, it is called incomplete. In terms of sexual reproduction in flowers, only stamens and pistils are necessary. Flowers with both structures are called perfect. But if they lack either one of the other, they are called imperfect.